In this video, we are going to study about the cerebellum. The cerebellum is the largest part of the hindbrain and the second largest part of the brain as a whole. It weighs about 150 grams and it is located in the posterior cranial fossa underneath the tentorium cerebelli. So this is the tentorium cerebelli. So it is present underneath the tentorium cerebelli and behind the pons and the medulla oblongata. It is separated from the pons and the medulla by a cavity of fourth ventricle. So this is the fourth ventricle which lies between the pons and medulla oblongata in front and cerebellum behind. When you see the surface of the cerebellum, it bears numerous fissures separating narrow folia. So this is the folia and this is the fissure. So the elevation is called as the folia and the depression is called as the fissure. So these are mostly transverse. The cerebellum is connected to the brainstem by three pairs of large fiber tract. They are called as the cerebellar peduncles. The three primary functions of the cerebellum are the maintenance of the posture, the maintenance of muscle tone, and coordination of voluntary motor activity. Next we'll see about the external features. The external features of the cerebellum consist of three parts, two surfaces, two notches, and three well-marked fissures. The parts are: the cerebellum consists of two large lateral hemispherical lobes, the cerebellar hemisphere, which are united to each other by a narrow median worm-like portion that is called as the vermis. The superior and the inferior aspect of the vermis are termed as superior and inferior vermis respectively. The ridge-like superior vermis is continuous on either side with the superior surface of the cerebellar hemisphere. The inferior vermis is more clearly demarcated from the hemisphere in the floor of the vallicula cerebelli. Next we'll see the surfaces. So this is the superior surface of the cerebellum. So the superior surface of the cerebellum is convex and the two cerebellar hemispheres are continuous with each other on this surface. This is the inferior surface. The inferior surface presents a deep median notch that is called as the vallicula, which separates the two cerebellar hemispheres. The floor of the vallicula is formed by the inferior vermis and it is limited on each side by the sulcus valliculae. Next we'll see the notches. The anterior aspect of the cerebellum is marked by a wide, shallow anterior cerebellar notch which accommodates the pons and the medulla. The posterior cerebellar notch is deep and narrow and this lodges the falx cerebelli. Next we'll see the fissures. The first fissure is the horizontal fissure. So this is a horizontal fissure. It is more prominent and runs along the lateral and the posterior margins of the cerebellum and it marks the junction between the superior and the inferior surface of the cerebellum. The second fissure is the fissura prima or primary fissure. So it is a V-shaped fissure on the superior surface. It cuts the superior vermis at the junction of the anterior 2/3 and posterior 1/3. And it divides the corpus cerebelli into anterior and posterior lobes. The posterior lobe is also known as the middle lobes. The third fissure is the posterolateral fissure. The posterolateral fissure lies on the inferior surface of the cerebellum and separates the flocculonodular lobe from the rest of the cerebellum. This is a schematic picture. This is the horizontal fissure. So this is the superior surface and this is the inferior surface. So next we'll see the various subdivisions or the lobules of the vermis and the cerebellar hemisphere. So the fissura prima divides the cerebellar hemisphere into anterior lobe and posterior lobe. So now we'll see the subdivisions in the anterior lobe. The superior vermis consists of lingula, central lobule, culmen. The cerebellar hemisphere has ala and quadrangular lobule, which corresponds with the central lobule and the culmen of the superior vermis. The lingula does not have any corresponding lateral projection in the cerebellar hemisphere. The posterior lobe consists of, in the superior vermis, you have declive, folium, tuber, pyramid and uvula. Correspondingly, in the cerebellar hemisphere, you have the lobulus simplex, the superior semilunar lobule, the inferior semilunar lobule, the biventral lobule and the tonsil. Finally, you have the flocculonodular lobe. So nodule and flocculus. So this forms the flocculonodular lobe. Again I repeat, so the subdivisions in the superior vermis and the inferior vermis are the lingula, the central lobule, the culmen, the decline, the folium, the tuber, the pyramid, uvula and nodule. The subdivisions in the cerebellar hemisphere are the alae or the ala, plura is the alae, the ala, then you have the quadrangular lobule, the lobulus simplex, the superior semilunar lobule, the inferior semilunar lobule, the biventral lobule, the tonsil and the flocculus. So this part forms the anterior lobe and this forms it's the posterior lobe and this is the flocculonodular lobe. Next we'll see the subdivisions of the cerebellum. Anatomical subdivisions. Anatomically, the cerebellum is divided into three lobes. The anterior lobe, the posterior or middle lobe and then you have the flocculonodular lobe. The anterior lobe lies on the superior surface anterior to the fissura prima. The posterior or the middle lobes lies between the fissura prima on the superior surface and posterolateral fissure on the inferior surface. The flocculonodular lobe is the smallest of all the lobe and it lies on the inferior surface in front of the posterolateral fissure. Next we'll see the morphological subdivisions. Based on phylogenetic and functional criteria, the cerebellum is divided into three parts: the archicerebellum, the paleocerebellum and neocerebellum. The archicerebellum is also known as the vestibular cerebellum. Phylogenetically, it is the oldest part of the cerebellum and the fishes and the lower amphibian possess only this component of the cerebellum. It consists of the flocculonodular lobe and the lingula. So lingula and the flocculonodular lobe forms the archicerebellum or vestibular cerebellum. The archicerebellum is chiefly vestibular in connection and it is concerned with the maintenance of equilibrium, tone and posture of the trunk muscle. Next we'll see the paleocerebellum or spinal cerebellum. Phylogenetically, it is the next part of the cerebellum to appear in the terrestrial vertebrates with the appearance of the limbs, example the reptiles and the birds. It consists of the anterior lobe except the lingula and the pyramid and the uvula of the inferior vermis. So the anterior lobe except the lingula, pyramid and the uvula of the inferior vermis forms the paleocerebellum or spinal cerebellum. The paleocerebellum is chiefly the spinocerebellar in connection, and they are concerned with the tone, posture and crude movements of the limbs. The third is the neocerebellum or cerebral cerebellum. Phylogenetically, it is the most recent part of the cerebellum to develop, and it develops in primates in association with the enlargement of the telencephalon and the cerebral cortex. The neocerebellum is made up of the middle lobe or the posterior lobe, which is the largest part of the cerebellum, except the pyramid and the uvula of the inferior vermis. The neocerebellum is chiefly cortico-pontocerebellar in connection, and it is concerned with the smooth performance of skilled voluntary movements. Next we'll see the internal structures. The cerebellum is made up of a thin surface layer of grey matter, the cerebral cortex, and a central core of white matter. Embedded within the central core of white matter are the masses of intracerebellar nuclei. So these are white matter. So within the white matter you have the masses of grey matter which are known as the intracerebellar nuclei. The cerebellar cortex is folded in such a way the surface of the cerebellum presents a series of parallel transverse fissures and intervening narrow leaf-like bands that is known as the folia. So these are the fissures and this is the folia. Each folium consists of a slender blanched lamina of central core of white matter covered by a thin layer of grey matter. The central core of white matter being arranged in the form of branching pattern of a tree, and it is called as the arbor vitae cerebelli. Next we'll see about the grey matter. The grey matter of the cerebellum is represented by the cerebellar cortex and the intracerebellar nuclei. Now we'll see about the structure of the cerebellar cortex. The structure of the cerebellar cortex is uniform throughout, that is homotypical. It consists of three distinct layers: an outer molecular layer, an intermediate Purkinje layer, and then you have an inner granular layer. So outer molecular layer, intermediate Purkinje cell layer and inner granular layer. The molecular layer or the plexiform layer is the superficial layer. It mainly consists of numerous dendritic arborization of the Purkinje cells, and there are relatively few nerve cells which are widely spaced. The nerve cells are two types. One is the basket cell, the other one is the stellate cells. The basket cells are small in size with little cytoplasm and extensive processes, and the longest of these processes is the axon of each cell, which assumes a transverse course which are parallel to the cortical surface and right angle to the longitudinal axis of the folia. the transverse axon, they synapse through numerous basket-like nets of collaterals with the dendrites of many Purkinje cells, about 500 Purkinje cells. Next, we'll see about the stellate cells. The stellate cells are characteristically short process and they are scattered near the surface. Their axons arborize with the dendritic spines of the Purkinje cells. Next, we'll see about the Purkinje cell layer. The Purkinje cell layer is the intermediate layer. It consists of single row of flask-shaped cells, the Purkinje cells. A dendrite arises from the neck of the flask and passes upward, and they undergo a profuse branching to form an elaborate dendritic tree. The dendrites of the Purkinje cells, they synapse with the collaterals of the basket cells, then the axons of the granule cells, then the climbing fibers. The axons of the Purkinje cell, they arise from the deeper pole and they pass through the granular layer and then into the white matter, where they relay with the intracerebellar nuclei, except those from the flocculonodular lobe, which passes directly to the vestibular nuclei. The outgoing Purkinje axons, they constitute the sole output from the cerebellar cortex and they exert an inhibitory influence on the intracerebellar nuclei. So the fibers are colored as red, so which are inhibitory to the intracerebellar nuclei. Next, we'll see about the granular layer. So this is the granular layer. The inner granular layer consists of numerous closely packed small granule cells, and this layer also contains a few large Golgi cells. Each granule cell gives rise to 4 or 5 short dendrites which make a claw-like endings which synapse with the terminals of the Morse fibers. The blue colored are Morse fibers, so they make contact with the dendrites of the granule cells. The axons of each granule cell pass into the molecular layer where it bifurcates at T junction and its branches run parallel to the long axis of the cerebellar folium, and these fibers are known as the parallel fibers. They run at right angle to the dendritic processes of the Purkinje cells and they make synaptic contact with them with the dendritic processes of the Purkinje cells. The Golgi cells are large and prominent but scanty, and their dendrites, they ramify in the molecular layer. So now we'll see about the cerebellar glomerulus. The cerebellar glomerulus is a complex synaptic structure consisting of following components. You have the rosette or the core of the glomerulus, which is formed by the termination of the Morse fiber. Then you have the dendrites of the granule cells, about 20 synapsing with the rosette. Then you have the axons of the Golgi cells synapting with the rosette and with the dendrites of the granule cells. The total number of neurons in the cerebellum are more than that in the rest of the brain. Next, we'll see about the intrinsic neurons of the cerebellar cortex. There are 5 types of intrinsic neurons of the cerebellar cortex. They are the Purkinje cells, then you have the granule cells, then you have the stellate cells, and then you have the basket cells, and the 5th one is the Golgi cells. All the intrinsic neurons of the cerebellar cortex are inhibitory except the granule cells. Such collection of inhibitory neurons is not found anywhere else in the CNS. exit in the cerebellum. Next we'll see about the intracerebellar nuclei. The intracerebellar nuclei, which is also known as the central nuclei, or the masses of grey matter embedded in the white matter of the cerebellum. On each side of the midline, they are 4 in number. From lateral to medial side, they are the dentate nucleus, the emboliform nucleus, the globus nucleus, and vestigial nucleus. The dentate nucleus is most prominent of the intracerebellar nuclei and largest in the primates, especially in humans. It is the nucleus of the neocerebellum, and therefore it receives the afferent fibers from the neocerebellum. In section, it has a shape like a crumbled bag appearance, with the hilum facing anteromedially. The interior of the nucleus is filled with white matter made up of efferent fibers that leave the nucleus through the hilum, forming most of the superior cerebellar peduncle. These fibers include the dentatorubral and the dentatothalamic fibers. From the dentate nucleus, you get the efferent fibers, the dentatorubral fibers, and the dentatothalamic fibers. The dentatorubral fibers, they end in the red nucleus. From there, the fibers project into the spinal cord. The dentatothalamic fibers, they end in the ventral lateral nucleus of the thalamus. From there, the fibers are projected into the cerebral cortex. The next nucleus is the emboliform nucleus. It is oval in shape and it is situated medial to the dentate nucleus, and it is partially covering its hilum. It is the nucleus of the paleocerebellum. Hence, it receives the afferent fibers from the paleocerebellum and gives fibers to the red nucleus via the superior cerebellar peduncle. The red nucleus projects to the spinal cord through the rubrospinal tract, which facilitate the flexor muscle tone. The next nucleus is the globus nucleus. It is rounded in shape and it lies between the emboliform nucleus and the vestigial nucleus. It has a similar connection to the emboliform nucleus, and the globus and the emboliform nuclei together sometimes referred as nucleus interpositus. The vestigial nucleus lie near the midline in the vermis, close to the roof of the fourth ventricle. It is smaller than the dentate nucleus but larger than the emboliform or globus nucleus. It is the nucleus of archicerebellum. Hence, it receives the afferent fibers from the flocculonodular lobe and convey the efferent fibers to the vestibular and the reticular nuclei. The vestigial connections influence the extensor muscle tone. Next we'll see about the white matter. The white matter of the cerebellum is made up of 3 types of fibers. You have intrinsic fibers, afferent fibers, and efferent fibers. The intrinsic fibers remain confined within the cerebellum and they connect the different regions of the cerebellum either in the same hemisphere or of the 2 cerebellar hemisphere. Through the afferent and efferent fibers, the cerebellum is connected with other parts of the CNS. Next we'll see about the connections of the cerebellum. First, we'll study about the afferent fibers. The cerebellum receives input through the afferent fibers mainly from the cerebral cortex, the spinal cord, and from the vestibular apparatus. The vestibular apparatus include the saccule, utricle, and the semicircular canal. In addition, it receives input from the red nucleus and the tectum of the midbrain. The cerebral cortex sends information to the cerebellum through the cortico-pontocerebellar pathway, the cerebro-olivocerebellar pathway, and cerebro-reticulocerebellar pathway. The spinal cord sends subconscious proprioceptive information to the cerebellum via the posterior spinocerebellar, anterior spinocerebellar and cuneo-cerebellar tract. The afferent fibers from the vestibular apparatus passes through the cerebellum either directly or after relay in the vestibular nuclei. The afferent fibers, they enter the cerebellum mainly through the inferior and the middle cerebellar peduncles, and they are classified into two types, climbing fibers and mossy fibers. The climbing fibers, they originate in the inferior olivary nucleus. Each fiber, after giving a collateral to the intracerebellar nucleus, make a monosynaptic contact with the Purkinje cells. The mossy fibers, they constitute the bulk of the afferent fibers of the cerebellum, and each mossy fiber, they end by forming 30 to 40 terminal swelling, that is called as the rosette. Each rosette forms a synaptic contact with the dendrites of the granule cells and axons of the Golgi cells. The rosette with the synaptic contacts with the dendrites of the granule cells and the axons of the Golgi cells is known as the cerebellar glomerulus. Each glomerulus is surrounded by a capsule of neuroglial cells, and they appear spherical or ovoid in shape. The climbing fibers synapse directly with a single Purkinje cell, whereas the mossy fibers, they synapse with the thousands of Purkinje cells through the granule cells. So one mossy fiber, they synapse with several granule cells, then each granule cell synapse with thousands of Purkinje cells through parallel fibers. Therefore, one climbing fiber specifically influences only one Purkinje cell, whereas the influence of mossy fiber is far more diffuse. The influences of both climbing and the mossy fiber are excitatory to the Purkinje cells. All the afferent fibers of the cerebellum are mossy fibers except the olivocerebellar from the inferior olivary nucleus and parolivocerebellar fibers from the accessory olivary nucleus are climbing fibers. Next we'll see the efferent fibers. The efferent fibers from the cerebellum passes to the red nucleus, the thalamus, the vestibular complex and the reticular formation. The entire output of the cerebellar cortex is through the axons of the Purkinje cells. Most of the axons of the Purkinje cells, they end by synapsing with the neurons of the intracerebellar nuclei. The axons of the neurons of intracerebellar nuclei in turn project to other parts of the nervous system. However, a few Purkinje cells neuron from the flocculonodular lobe and the vermis, they pass directly out to the cerebellum to end in the lateral vestibular nucleus. Fibers from the dentate nucleus, the emboliform nucleus and the globus nucleus, they leave the cerebellum through the superior cerebellar peduncle, whereas the fibers from the fastigial nucleus leave the cerebellum through the inferior cerebellar peduncle. Next we'll see the intrinsic neuronal circuitry in the cerebellum. All the afferent fibers to the cerebellum, example the mossy fibers and the climbing fibers, are excitatory to the cells of the cerebellar cortex, and their collaterals are excitatory to the intracerebellar nuclei. The climbing fibers, they excite the Purkinje cells directly. So green color arrows are excitatory, whereas the red color arrows are inhibitory in action. The climbing fibers, they excite the Purkinje cells directly. On the other hand, the mossy fibers, they excite the Purkinje cells indirectly through the granule cells. The granule cells, in addition to the Purkinje cells, also excite the basket and the stellate cells. So these basket and the stellate cells, they inhibit the Purkinje cells. So this is known as the neural sharpening mechanism. The mossy fibers also excite the Golgi cells and they provide the feedback inhibition to the granule cells. The Purkinje cells exert an inhibitory influence on the neurons of the intracerebellar nuclei, which in turn they modify the muscular activity through the motor control areas of the brainstem and the cerebral cortex. Next we'll see about the cerebellar peduncles. The afferent and the efferent fibers of the cerebellum are grouped together on each side into three large bundles. They are called as the cerebellar peduncles. The superior cerebellar peduncle connects the cerebellum to the midbrain. The middle cerebellar peduncle connects the cerebellum to the pons. The inferior cerebellar peduncle connects the cerebellum to the medulla oblongata. The inferior cerebellar peduncle is also called as the restiform body, and it is formed on the posterolateral aspect of the upper half of the medulla oblongata. The two peduncles, they diverge as they ascend and pass into their respective cerebellar hemisphere. The inferior cerebellar peduncle consists mainly of afferent fibers to the cerebellum from the spinal cord, the olivary nuclei, the reticular formation of the medulla, and the vestibular nuclei and the nerve. It also transmits the efferent fibers from the cerebellum to the medulla, principally the vestibular nuclei and the reticular formation. So next we'll see the afferent fibers of the inferior cerebellar peduncle. So the first fibers are the posterior spinocerebellar fibers. These fibers arise from the ipsilateral thoracic nucleus or Clark's column. The second fibers are olivocerebellar fibers. They arise from the opposite side inferior olivary nucleus. The third group of fibers are the parolivocerebellar fibers. They arise from the opposite side medial and dorsal accessory olivary nuclei. The fourth group of fibers are the posterior external arcuate fibers or cuneo-cerebellar fibers. So these fibers arise from the ipsilateral accessory cuneate nucleus. The fifth group of fibers are anterior external arcuate fibers. They arise from the arcuate nucleus of both sides. The sixth group of fibers are the vestibulocerebellar fibers. They primarily arise from the vestibular nerve and secondary from the medial and the inferior vestibular nuclei. So these fibers form the juxta-restiform body. The seventh group of fibers are the reticulocerebellar fibers. They arise from the lateral and the paramedian reticular nuclei of the medulla oblongata. The efferent fibers which are present in the inferior cerebellar peduncle are the cerebello-vestibular fibers. So these fibers arise from the ipsilateral flocculonodular lobe and the vestigial nuclei of both sides, and these fibers passes through the juxta-restiform body. And the second group of fibers are the cerebello-reticular fibers. They arise from the vestigial nuclei of both sides to the pontine and the medullary reticular formation. The third group of efferent fibers are the cerebello-olivary fibers. So they connect the cerebellum with the inferior olivary nucleus. Next we'll see about the middle cerebellar peduncle, the afferent fibers which are present in the middle cerebellar peduncle. So first is the pontocerebellar fibers. They form the main bulk of the middle cerebellar peduncle, and these fibers arises from the pontine nuclei. They run transversely and cross the midline and enter the opposite side cerebellar hemisphere, that is the neocerebellum. The pontine nuclei receive input from the cerebral cortex, so thus forming the cortico-pontocerebellar pathway. The second fibers, afferent fibers, are the reticulocerebellar fibers. They arise from the reticular formation of the brainstem of the same side and are projected to the vermal region of the cerebellum. The third group of fibers are the ceratogenic fibers. They arise from the raphe nuclei of the pons. The middle cerebellar peduncle does not have the efferent fibers. Next we'll see about the superior cerebellar peduncle. First we'll see the afferent fibers in the superior cerebellar peduncle. These include the anterior spinocerebellar tract, then the tectocerebellar fibers which arises from the tectum of the midbrain, that is the superior and the inferior colliculi of both sides. Then the trigeminocerebellar fibers, they arise from the superior sensory and the spinal nuclei of the trigeminal nerve. Then the cerebellocerebellar fibers which are noradrenergic fibers from the locus coeruleus. Then hypothalamocerebellar fibers which are cholinergic fibers from the hypothalamus. The efferent fibers which are present in the superior cerebellar peduncle include the cerebello-rubral fibers. They mainly arise from the globus and the emboliform nucleus, and they project to the contralateral red nucleus. Then you have the dentatorubral and the dentatothalamic fibers which arises from the dentate nucleus and project to the opposite side red nucleus and the thalamus. Then you have the cerebello-olivary fibers which arise from the dentate nucleus and passes through the opposite side inferior olivary nucleus. Then you have the cerebello-reticular fiber which arises from the nucleus fastigius and they passes through the reticular nuclei. The cerebellum plays a key role in accomplishing smooth and coordinated movements by means of its comparator function. If the cerebral cortex decides on a movement to do, the impulses from the motor cortex stimulate the anterior horn cells, that is the lower motor neuron of the spinal cord, to initiate a voluntary movement, and at the same time the motor cortex tells to the cerebellum about the intended movement. The cerebellum also receives the proprioceptive information from the muscles and the joints, telling it what movement is actually occurring. The cerebellum compares the intended movement with the actual movement occurring, and if a difference is detected, the cerebellum sends the information to the motor cortex and the lower motor neuron to correct the discrepancy. The result is smooth and coordinated movements. With training, a person can develop highly skilled and rapid movements, and the cerebellum thus plays an important role in learning of motor skills. Next we'll see the arterial supply of the cerebellum. The cerebellum is supplied by three pairs of cerebellar artery. The superior cerebellar artery, then the anterior inferior cerebellar arteries, then the posterior inferior cerebellar artery. The superior cerebellar artery is a branch of the basilar artery and supplies the superior surface of the cerebellum. The anterior inferior cerebellar artery, a branch of basilar artery, supplies the anterior part of the inferior surface of the cerebellum. The posterior inferior cerebellar artery, a branch from the vertebral artery, supplies the posterior part of the inferior surface of the cerebellum. Next we'll see the clinical correlation. The cerebellar syndrome. The cerebellar lesion due to the trauma or vascular occlusion, tumors, etc. produces a number of signs and symptoms which together constitute the cerebellar syndrome. The signs and symptoms produced by the cerebellar lesions are as follows. The involvement of the archicerebellum will lead to the truncal ataxia and staggering gait. The patient is unsteady while standing and staggering gait while walking. He walks with legs well apart, that is on the wide base, and sways from side to side. And next you have the nystagmus, the involuntary to and fro oscillatory movements of the eyeball while looking to either side. The patient is unable to balance while standing upright and falls on closing the eye, that is known as the Romberg sign. Involvement of the paleocerebellum will lead to diminished muscle tone, that is hypotonia of the limb muscle. This manifests in the form of lack of stability of the joint. Then you have the disturbances of the tendon reflex, namely the tapping of patellar tendon produces oscillating movement of the leg swings backward and forward. This is called as the pendular knee jerk. Then there is an inability to maintain the balance while walking, that is known as ataxia. Involvement of the neocerebellum leads to muscular incoordination, that is called as asynergia, and this manifests in the form of dysmetria, that is inability to measure the distance correctly for reaching an intended target, described as past pointing. Clinically, this is tested by finger-nose test. The patient is asked to touch the tip of the nose by his finger. He either undershoots or overshoots the tip of his nose. When the patient tries to correct the mistake, it results in to and fro movement. Then you have the intentional tremor, probably due to dysmetria. The tremors occur during purposeful movements and disappears with rest. Then you have adidadokinesia and dysdiadokinesia, that is inability to execute alternate movements in rapid successions. Example, pronation and supination of fora. Then you have dysarthria or scanning speech. The speech is slurred, prolonged, explosive, and with pauses in wrong places. The fifth feature is the rebound phenomena. The patient is unable to check the action of the agonist muscle by the corresponding antagonist muscles. If the patient is asked to push the palm of the doctor by his palm, the force that the patient can develop slowly rises, and now if the doctor suddenly remove his hand, the limb of the patient flies back, that is rebounds and hits against his chest because he is not able to stop his pushing act immediately. The lesion of the cerebellar hemisphere give rise to the signs and symptoms on the side of lesion, as each cerebellar hemisphere is connected by nervous pathway with the same side of the body. It is important to know that the cerebellum and the basal ganglia both control motor function but have opposite effect, and many signs and symptoms associated with their dysfunction are also opposite. For example, the cerebellar dysfunction results in decreased muscle tone and an intentional tremor, whereas the dysfunction of basal ganglia results in increased muscle tone and resting tremors.